For decades, aging and mortality was seen as fate. We now know it's a biological process that can be slowed and perhaps even reversed. But what if the key to next generation longevity science isn't hidden in our cells, but in the laws of physics? Welcome back to Decoding Longevity, where we explore the frontiers of longevity science and technology that are shaping our health. I'm your host, Raghav Sehgal, and with my co-host, Macsue Jacques.
Hello, everyone. In our last episode, we talked about DNA methylation and how we can use it in multiple ways to draw important biological insights based on the way that these tiny chemical marks are changing with aging. And to build on some of the concepts that we started to touch on in the last episode, we are joined by a very special guest, physicist, biotech entrepreneur, and longevity pioneer, Peter Fedichev, founder of Gero, one of the few companies taking a radically different physics-first approach to understanding human aging. So, Peter, you have a fascinating background, going from theoretical physics to aging research and biotech. Can you walk us through that journey? And more specifically, how does your physics training shape the way you think about aging and biology?
Thank you very much for having me first. And second, yes, I was interested in science since I was a schoolboy, and I couldn't decide between actually physics, chemistry, and biology. So I kind of evolved, started from chemistry, evolved over the scales of nature, touched biology somehow. Well, I'm Russian, so in Russia, as you may know, there was a very strong school in theoretical physics. So when I started approaching mentors, they basically told me the story that there were physicists who moved to biology, but they told me that there were no biologists that went back to physics. So basically, they said, young man, shut up, take the physics textbook and go study to a good place. Actually, it was not true. So since then, I learned that there is at least one very fascinating example, Stuart Kaufman, the guy who influenced my switch, actually. So he is a neuroscientist and a doctor, a medical doctor, who turned to be a very strong theoretical physicist. So that's to the end of this story. So I was doing theoretical physics. I like to say that I used to have a decent job. I was doing pretty good problems. I was working with very strong people. And if you think about physics over the last 20 years, I think you would agree with me that imagine somebody in 2005, let's say, you could do string theory, you could do quantum computer, or you can get depressed because as you know physics lives in a nightmare scenario. In biology in biology of aging we have hundreds of theories of aging, right? And in physics we have a standard model which is definitely not a theory because it's wrong, but it has only two wrong predictions with an accuracy one in 10,000 or something like that. So fast forward 20 years now, string theory is dead, quantum computer still doesn't exist in any practical way. And happy me, I switched to biology of aging.
Well, I'm glad that you made that switch. And that's a really interesting story, I have to say. I will say I have a very similar history as well of switching from electronics engineering to biology. So I can relate to some of the interesting sort of choices that you made. But even within aging, I think we're in a very interesting time where I would almost argue it's kind of like the golden age of aging research. There is so much money flowing, billions of dollars flowing into sort of these really massive companies, startups, potentially a lot of new molecules. And yet, I believe that we don't have really a reliable way of slowing down aging in humans. Well, other than maybe calorie restriction, right? So, and sort of this real progress that we're looking for towards sort of these exploring new anti-aging therapeutics has been unfortunately frustratingly slow. Why is that, Peter, in your opinion? What are we missing?
Well, of course, you can only answer this question once you create a drug that extends human life by 200 years. Everything else is a speculation. And another bad joke in our field is that when do you think a drug that would extend human lifespan by 200 years would be created? And the correct answer is unfortunately in 200 years, because you have to you have to test that. So that makes us in a very precarious situation trying to discuss it, right? So infinite number of people have infinite number of ideas why this happens. So Well, I have this kind of, physics is engineering on steroids, I would say, because physics is a very practical science. And when you cross to biology, you see a number of cultural, I would say, barriers. And some of them, I think, actually prevent us from developing a drug, a strong intervention against aging. So I will try to clarify that. So to me, physics is an interesting science because it has very strong theoretical, whatever, mathematical foundation, not statistical, but mathematical foundation. So in physics, you can model. And the level of modeling in physics is such that you can ask your government to spend $10 billion on nuclear weapons, even though nuclear weapons do not exist in nature. So biology is, in a way, on the other side of this spectrum. So in biology, culturally, everything is perceived complex. Modeling is useless. Statistics is all powered, which means that you are limited to things that exist in nature. So you are trying to explain what exists in nature. And the problem with that is that, look, actually, in science of aging, we're in a better situation than in biology on average. Because what contributed to my switch is that there were suspicions that certain animals do not age or age very slowly, right?
Yeah.
And before that, we are epics of creation, God chosen, species, and of course, we're the best, and we're aging. So if we are aging, so how can you dare actually think about stopping aging, right? So fortunately, like counter to this example with nuclear weapons, fortunately, we have running examples, many species that presumably do not age, right? So at least we know the answer that there are certain animals that intercepted aging. We know that there are many of them. So I call this criminal negligence in science, right? So I think people have been studying aging for, I don't know, let's put it generously, 10,000 years. I think a lot more. And then in 2006, you can publish a paper on a mammal that presumably does not age. And you cannot publish it in Nature. You have to publish it for Google money, right? I mean, for this totally nonsense work, you cannot get a grant, right? I mean, really totally non-interesting science, right? You have to build Google, earn tens of billions of dollars, set up a daughter company, Calica, that will finally find $500,000 in order to check this totally uninteresting fact. So back to this, we have now this running example of species that presumably do not age. They are not hydras, these are mammals, there are bats and other things. So if you publish this paper in 2006, think how many other animals do not age. And we just don't know, because to establish that something doesn't age, you need thousands of years. So if the field is so nascent that you can make this level of discovery in 2006, maybe it's a good time to switch from another field. That's kind of my short answer.
Yeah.
That makes a lot of sense. Yeah.
That is great. So I think like just moving on and touching on like the how we started to study aging. We know that you mentioned 2006, and alongside that we have like a overflow of data. So you have the creation of all the other omics and all these different approaches, which is generating like so much data. But I think the issue here, and please correct me if I'm wrong, is that it's not that we don't have enough data to move on the field, but we actually lack a unified approach, or we lack a unified measure. And from my research and basically reading what you have published, could you say that this measure is entropy, or is that the same thing when you say that you're talking about aging, loss of resilience in biological systems? And what does that mean in simple terms? Let's start there.
Yeah, I think we can touch this a little bit philosophically. I think that biology, and especially molecular biology, is culturally a very reductionist science, right? So you are publishing papers that gene A causes B, C, and D, and that causes diseases, and that's why let's hit A and cure the disease. So that basically tells you that you can achieve molecular level of control on complex things like aging and chronic diseases. And to an extent, it works. So if you have an acute situation, you can actually find, if a virus attacks a human body, you can actually find a viral protein, hit it with a drug, and the virus disappears in a few hours. So that works. But unfortunately, in complex systems that evolve over long times, most of details of interactions between individual parts don't. I mean, it doesn't matter if I slept today well or not for my lifespan. And there is a lot of molecular level information in me right now that is concerning if I was sleeping well or not. So in a way, we are cursed by the amount of information that we have right now. And that's where I think physics is very important, because, I mean, to predict my next word, you really have to measure a lot. on me. But to predict my time of death, in a way, you don't need so much information because most probably my second date of my grave does not depend on the character of this conversation. Well, I hope it depends because it brings attention to the to the science of aging and eventually will contribute to the creation of a drug. But this is very, I mean, think how much that data you would need to collect about me and society in order to arrive to this conclusion, right? So physics used to be in this situation, and physics had to explain steam engines at the time when people were not sure that molecules exist. So culturally, physics was pressed in order to develop this kind of microscopic, self-averaged control quantities that describe complex systems at large scales. To the extent that you don't even care what kind of fuel is burned in your steam engine, you can do predictions, you can compute power, you can compute all the engineering quantities using very rough measures, which are at the same time are the most useful. So what I think needs to happen is that when you have so many variables, you cannot run so many statistical tests. Think about this, whatever information criteria, everything that people are taught in the statistical courses. This doesn't work if you have complex models like modern machine learning and lots of data. You have to aggregate them. And physics learned how to aggregate complexity to a few useful quantities. And you can actually do modeling, you can do predictions. And once you understand what's going on, you open yourself a way to create strong therapeutics. That's kind of my reasoning of the
Yeah,
challenge.
that's a very interesting insight, I would say, because it's very counterintuitive in some ways to how, as you pointed out, modern biology works, where we're like typically knocking off one gene or sort of manipulating a different protein to actually see how does that affect maybe a few other genes or proteins and the system in general. It's very counterintuitive and maybe might actually ruffle a few feathers with biologists, I would say. But I want to come back to this point around what you mentioned is specifically how do we go about modeling sort of such complex amount of information into some very key variables, right? Of course, there's mathematical and computational approaches that could do that, but how are you thinking about this problem
Yeah,
specifically?
I will try to throw a few things at you because, as I said, physics faced complexity, I think, a little bit earlier than other sciences because it took a while for biologists to learn how to collect those massive amounts of data. And even large biobanks, they only exist for, I don't know, 15 years, right? So this is all new. So I think if you think about public perception of physics, I think most of the people know that something like quantum mechanics happen, right? I mean, there is a public discourse on quantum mechanics, There is also a public discourse about general relativity. So I think this is kind of part of the common cultural narrative right now. But if you think about Nobel Prizes in physics in, let's say, 70s, like Nobel Prizes for phase transitions in the randomization group, they haven't yet penetrated any cultural domain. And this information doesn't diffuse to other sciences. So, for example, Eugene Koonin published mass paper in biology and randomization group and evolution just last year, right? So there is a certain gap. So what happened there in physics in the 70s, I think it's very interesting and super relevant to biology. So physicists started thinking what happens if you go up in scale, right? And physicists started thinking what happens when you go from one scale to the other, right? Because when you go from one scale to the upper scale, you do a lot of averaging. Those who study statistics know that there are very strong theorems like whatever central limiting theorem that tell you that most of the information disappears when you go up. And only very few variables survive averaging. Right, so that was a discovery that led to essentially randomization group. The observation was that the more you move up in scale, the more relevant variables are required to describe the system. And most of the time, systems get simplified on a larger scale. Information doesn't flow up when you go up in scale. Most of the microscopic information turns out to be irrelevant. That's the curse of drug discovery. As I told you, if you do biology on the same scale as the disease, as an acute viral infection, you are super successful. And success of your clinical trials is infinite because you do experiment in a dish and an animal on the same time scale. If you learn something, you learn something. You go to humans and it works. But once you start treating, for example, diabetes, that takes, I don't know, 10 years, 10 times more time to develop in mice than in humans, you go up in scale and you lose information. Nothing works, right? So, which means that without using special tools and philosophy, ideology that forces you to identify those relevant, not self-averaging quantities that are biological age, frailty index, and everything else, and establish relations between them, you are just failing, because nothing that you learn on a microscopic scale or on the scale of hours and days actually help you to develop a drug. I mean, that's why we fail so much in clinical trials. If we don't understand, this is called hierarchy of scales, and the great paper in Science about this is Phil Anderson, one of the greatest theoretical physicists of late times, and it's called More is Different. Guys, more is different, and you have to learn how to go up in scales, and physics learn that.
Yeah, I think what you touched on is a little bit of with the Gero approach, right? So I think, in my opinion, and I believe Raghav as well, it's quite incredible how you managed to study aging in such a dynamic way, like bringing things from the 1970s to what we facing now with all this data. So let's dive in a little bit more. So you said that you're modeling aging not as a collection of diseases, but everything is failing, like things just started failing. And I think this is what you refer to as a thermodynamic process. Do you explain a bit more?
When I came to the biology of aging, I thought, well, the theoretical physicist way to look at aging would be like this. Like, you can have most probably two limiting cases, right? I mean, in one case, there is one system that is responsible for aging, right? I mean, kill it, live forever. I have infinite respect to biologists and medicine and medics, meaning that if such system existed in humans, I mean, maybe it's different in all animals, but if such system existed in humans, it would have been removed by surgery already, or by mutation in some, or a few successful individuals in some, family clan in a remote village that would be controlling the world by this time. So this is not what we observe, right? The same, we actually see that aging is ultra robust with respect to mutations, for example, right? No amount of lifestyles, yoga, spiritual practices, bad or good food moves the needle in terms of lifespan by any reasonable number, right? So if you reduce the effect of diseases, which is important, right? Because people having better access to medicine live longer in the wrong shape. If you look at the actual progress or evolution of maximum human lifespan, you see that there is nothing, I mean, really, literally nothing that we can do to us with all the arsenal of modern technology, including bad stuff. I mean, think about smoking. There is nothing worse than smoking, right? But even this is just, slightly larger than 5 years in lifespan, which is like 5 over almost 100. I mean, we're talking about few percent effects, which and it's literally difficult to kill yourself other than by absorbing an acute poison. I think this is very, insightful, that hints you that aging is not one process, right? Because this is the intuition that comes from physics. Assume that nothing, I mean, think about random perturbations that you can throw at a human. Then, on average, they have effect zero, right? Intervention to intervention. Some of them kill you, some of them extend your life. But the problem is that all of them change your lifespan just by a very small number, meaning that most probably they affect multiple systems that are responsible for aging in a random way each. And that's why statistical theorems are telling you that you average to zero.
Yeah.
So most probably that means that on this kind of continuum, aging in humans is not one failure, but many. And that means that this mechanistic or reductionistic approach would not work in humans, specifically for that reason.
Yeah.
And, I can experimentally try to explain you what is the physicist kind of intuition in the other limit.
Yeah, actually,
But
I
you
have a
can tell.
question on this front, maybe, which is like, this is when we are talking about this reductionist approach. Would you say like these biological age clocks and predictors are then oversimplifying things a bit? Do you think a single biological age measure is enough, or do we need sort of these almost multi-omic clocks? But then that goes back to the point of using multi-omic data.
I can speculate on that as well. So I always like to answer these questions from empirical observations. And empirical observation is that hospitals have many departments for some, let's say, anecdotal reasons, meaning that the end life trajectories of humans are different. So humans start similar, but at the end of life, they are very heterogeneous, which means that if we want to maintain life, at the end of life, we have to have experts of different profiles in the medicine. And each of them comes with their distinct set of measurements. So let's say empirically, it means that at the end of life, we are different and we have theta. I mean, the closer we are to death, the more measures we need to describe. And this is, by the way, fits well into this idea of this intuition of complex systems. Complex systems under good control are described by few variables.
Yeah.
And systems that are disintegrating require a lot more variables to get described. And those variables are increasingly useless at the same time. So basically, it means that if you want to keep human alive at the end of life, which is not actually countering aging, that's something else, right? So basically, the closer you are to death, the more complex is the control problem. You need to measure more parameters and you have to control them in an increasingly more accurate way. So this is actually the control catastrophe, right? Which is very much related to the second law of thermodynamics. If we instead start concerning ourselves about aging, not individual diseases, then we're in the other limiting situation. So aging is a single process. When it starts, it causes diseases. The more diseases you have, the more variables you need. And this, by the way, is the intuition already in the field of aging, because people got this first generation clocks that essentially we're measuring chronological age. And this is close to what you would think aging is. And then some smart people, Morgan Levin and Steve Howard, came with the other clock, like Dream Age, saying that let's predict the age of death. And of course, they just make a clock that measures cardiovascular system health. And of course, if you want to make it accurate, you would like to add other systems there. And the problem is that those systems are not correlated anymore, because you can have good cardiovascular health, but poor liver, for example. And that's why you would have to have a liver clock, and you would have to have a cardiovascular clock, and you would have to have a brain clock. You will have to build infinite number of clocks. The issue here is that if we want to, the more variables you need, the shorter is your horizon of prediction. So aging, by definition, is the thing that is predicting your second date on the grave, which is kind of long-term prediction. And that thing is actually the most collective variable that exists in your body. And that's why if we really want to keep us physically well and living long, we have to focus on the period of life where those measures are few and concentrate on the one that corresponds to aging.
Now, I think what you touched on in a very, very key aspect of aging definition, because as you said, us scientists, we do consider aging as, the major disease per se, because it precedes everything, right? It's leading to everything else. However, for a standpoint where we doing research or we applying for grants or we're trying to come up with a drug, I would say aging is not considered a disease. So that kind of becomes a crossroad where we can't target aging per se, but we have to target disease by disease, which is not really sustainable, right? And I think one thing that I always question in my data is I'm looking at all these different issues and, is there a common measure? But one thing that I always come back to my mind is there a critical point that we can identify that is common in an individual or common to aging, where if you identify that point, you then don't diverge into this complexity and start having all these measures? And could you then intervene in that point to then potentially extend lifespan or health span?
You touched, I think, on a very important thing. If you follow my logic, which could be wrong, but for the sake of the conversation, if we keep pushing, my proposal is that I just told you that by, I think, empirical observation, we see that aging is not a single process. And this is an entropic catastrophe, right? If you have many, it's just either one or two, and then we can make two drugs and, we're living forever. Or alternatively, there is a situation where these are many processes. And most probably these processes are not even correlated to each other, right? So you would ask me then, what is the hope, right? If smoking doesn't kill you, how can we dare to think that something can actually stop that? First, there are animals that presumably do not age, so think harder. There are examples. We know that they arrive to this no aging in different ways. I like this comparative biology papers on bats. 1/3 of families of bats are not aging. And if you look at their genetics, you see that there is more than one way to arrive to that phenotype. Okay? This is already pretty encouraging. You don't need to get small and learn to fly in order not to age, right? So I think this is very encouraging. You start thinking this phenotype, non-aging exists, and it mechanistically can be generated in different ways. So you start thinking, how can you possibly do that? I will just throw two ideas at you, how this can possibly work, but also in your data. So assume that this is this entropic soup, like you have many processes that independently generate whatever damage that is accumulated and effectively kill you. So, of course, each of such damage events has some effect on your health. Some of them could be even beneficial, but on average, they are not beneficial because otherwise evolution would drive us, would simply use all those beneficial states and we would be living longer by default. Most of them are bad and they add up. Right, so the more you live, the more these damaging events you have. So that's why they build up. The effect of each of them on your health is nothing. If I have some, whatever, mutation from the left cell in my liver somewhere, it doesn't matter. But since there are many cells and everything else, the background of these things is going up. And this is called, in physics, this is called mean field, this very interesting concept. So there is a property, which is roughly the total amount of damage in your body, that is not microscopic, right? Because it's not located anywhere in your system. But the more damage you have, the more it has effect on every cell, right? Because the effects of all damaging events add up.
Yeah.
So each damaging event does nothing to the sum. But the sum of the damaging events helps new damaging events to appear. So in this way, this background, this dark matter of errors, each of them is benign, right? I mean, none of them individually can kill you. It doesn't exist. It's not localized anywhere microscopically. You cannot get a part of the system and look, where is this mean field of this damage? But it exists microscopically as a microscopic property of the system and defines your resilience, lifespan, and everything else. So this is the example of emergence, by the way. So this property is not microscopically controlled, but it's microscopically, it's very important, right? So you need to find ways to identify those microscopical properties. And then, since we know bats, naked mole rats, and everyone else, learn how to control them. So you can do comparative genomics of different species. You can do genome attestation studies on those quantities. And you can try to find if there is any genetic variation, for example, that is controlling these things. This sum is a very natural, the total amount of damage is a very natural biological age rate. And physics, everything is simple, right? So if you find such a microscopic quantity, it always has a simple meaning. And then you can bridge the microscopic to microscopic divide by going to the molecular level data and trying without suspecting any mechanisms. Try just to find which measures control the rate of accumulation of the thing, and you can possibly find it. In short, you cannot build this from ground up. This is called emergence. Emergence does not allow you to go from reductionist models up, right? Because the information doesn't flow up in complex systems. But you can measure relevant quantities on the upper level, and you can correspond it statistically with the bottom level. You can read the answer in the end of the textbook. This is an interesting example where you cannot solve a problem like in a textbook, right? You cannot go up, but you can look up.
Yeah, no, that's really interesting. And actually, I have a lot of follow-up questions here, but we're going to take a break on this episode, continue to explore and interview Peter in part 2 of our podcast around physics of aging. So make sure to join us as we continue this exciting discussion with Peter on the next episode. And as always, if you like the content on this podcast, make sure to subscribe and like, as well as check the show notes for some of Gero's papers and links for few bonus reads. Thank you for joining us at Decoding Longevity podcast. See you next time with some more exciting science.